We are thrilled to have with us today Dr. Nandita Mitra. She's a professor in the Department of Biostatistics, Epidemiology, and Informatics at the University of Pennsylvania, and she's co-director of its Center for Causal Inference. Her primary research interests include the design and analysis of observational studies, causal inference, and statistical approaches for cost-effective analysis. The title of her talk is Interpret with Caution, Drawing Conclusions from Real-World Data, and she is going to present about some of the concerns or some of the good uses and the cautionary uses of real-world data. And we will hear from her for about 20 minutes and then have time for Q&A, time for questions. If you want to ask a question, raise your hand, your Zoom hand, and we'll call on you, or put it in the Q&A text function. So Dr. Mitra, I want to thank you very much for joining us, and I'll turn it over to you.
Great, thank you so much. I'm delighted to be here. I am a statistician. I'm a professor of biostatistics at Penn. And in my other roles as co-director for the Center for Causal Inference and also the editor-in-chief of the journal Observational Studies, I think a lot about how to draw conclusions from real-world data. And so today I'll just give you a statistician's viewpoint about the drawbacks of using real-world data and where you need to be cautious, and different instances of where you need to be careful about making specific types of inference. Oops. See if I could change it. So I know earlier in the day, you've heard a lot about electronic health records, one type of large observational database that represents real world data. And there are many advantages to using EHR. There's no need for patient recruitment or data collection. So for example, in a randomized trial, it has to be very carefully designed. It could take years to run. It's very expensive. Here, these data are naturally collected by clinicians and are stored in health databases. And so the advantage is that you have access to a very large amount of data, something that we statisticians love. And it also allows us to look at a very diverse population. So clinical trials oftentimes are specific to a particular group. They might exclude older patients or patients who are more sick. But here we have access to all patients who are being treated within particular health systems. So it allows for a diverse population of study, and it's potentially more generalizable than a clinical trial. And there is potential with some challenges of combining data across multi-sites. So that's where statisticians and informaticians and computer scientists get together to really try to merge lots of data sources from different health systems. So in effect, it's a large amount of diverse data that potentially are easy and cheap to access. On the other hand, what we do worry about oftentimes is the data quality, and the quality could be poor. So there's the trade-off between vast amounts of data and the quality of the data. So for example, there could be missing data that are actually informative in the sense that, some patients who are more sick may be going to see their doctor more often. So there's more data on those kind of patients, or those patients who worry much more about their health, so go in very regularly. We have more data on those patients, but much less information and data on other patients that are in between those two extremes. So there are issues with quality, missing data, and also many of the notes that are taken are text notes, and those may be in the EHR. So we have to have pretty fancy methods to do text mining, to be able to glean information from those texts, and those can be very error prone. So there are both pros and cons. But what I'd like to do today is point out 4 different areas where I found that in newspapers and in the media where there may be some misinterpretations. Specifically, I'd like to talk briefly about misinterpreting probabilities. I apologize that I'm one of the last talks, and I'll be talking about probabilities. I hope that it's not too boring. I'd also like to talk a little bit about the danger of making causal claims from observational databases. So what does it actually mean to say that something causes something else versus an association? I'd like to point out a couple of instances where the original database, the original real-world database had major issues that actually led to publications that were retracted. So I'll mention a couple of those. And then finally, I'll end with a couple of instances where there was a lot of cherry picking of studies and also some reporting bias. So I'd first like to start talking about probabilities. So on the right there, you see a tweet from the Wall Street Journal. This was from just a few weeks ago they said about half of adults infected in an outbreak of the Delta variant of COVID-19 in Israel were fully inoculated with the Pfizer vaccine, prompting the government to reimpose an indoor mask requirement and other measures. So why is this somewhat problematic? Because that half of adults, that 50%, sounds very alarming. But that's really getting at that first probability that I have on the left there, where that vertical line is saying given. So what I'm writing out here in math shorthand is the probability of being vaccinated given that you're infected. So basically what this Wall Street Journal tweet is saying is amongst those who are infected, half were vaccinated. But that's not necessarily what we're interested in. What we want to know is the second probability that I have there, which is given that you're vaccinated, what's the probability that you could get infected? That's what we care about more. And those two probabilities, those conditional probabilities, are not equal. And there's a lot of misinterpretation of probabilities because these two things are switched around. So again, we're more interested, I would imagine, in the probability of amongst those who are vaccinated. If I'm vaccinated, what's my chance of getting infected? So a little bit more on conditional probabilities. There's also been some reports in the media saying that there's this result, which seems very paradoxical, of over 50% of COVID-19 deaths being among the vaccinated. So what we really want to think about is these four different conditional probabilities that I've written out. So the first one, given that you're old, what's the probability of dying? That's high. Given that you're old and the probability that you're vaccinated, that's high. On the other hand, the probability of being young and dying is low. And the chance of being young and being vaccinated in this country right now is lower. So what this is showing is that once you combine these probabilities, if you combine the old and the young together, you can end up with something called Simpson's paradox, which is this really strange paradoxical result of greater than 50% of COVID-19 deaths are amongst the vaccinated. So the key here is that age is a really important factor. It's a factor that's both associated with being vaccinated and with the outcome. So it's called a confounding variable. So something that we should think about carefully, and we as statisticians think about carefully. So one more slide on conditional probabilities, and it's just to introduce the concept of Bayes' rule. So again, in 2020, last year, some were claiming that COVID infection presents a serious hospitalization risk for young people. So to support this claim, they argued that a high proportion of those who were hospitalized were young. Okay, so look at the direction of that probability. So here, let's let A be someone who's hospitalized, B be that you're young. So then the hospitalization risk amongst young people is the probability of A given B. Given that you're young, what is the probability of being hospitalized? But that's not the same as the probability of B given A, which is saying that a high proportion of hospitalized patients are young. So here, Bayes' rule is just showing that the probability of A given B is equal to the probability of B given A times these two other probabilities. So we have the probability of A, which is the probability of actually being hospitalized in your population. And the probability of B is the proportion of people who are young. So these two probabilities are not equal. And it really does depend on those two rates, the proportion of people who are hospitalized and the proportion of people who are young. So using Bayes' rule, it's really easy to see that those two conditional probabilities are not equal. Again, I've seen this a lot in a lot of studies where those two probabilities are confused. So the next point I wanted to make is about when we can make causal claims. And so I know you've heard a lot about VAERS, and we've read a lot about VAERS. So just briefly, I wanted to mention a couple of pros and cons, and then talk about the issue of causality. So VAERS, of course, is the vaccine adverse event reporting system that was developed by the CDC and the FDA about 31 years ago, when most citizens didn't have access to the web. And so it was purely a reporting system for clinicians and pharmaceutical companies. Now, since anyone has access and can report events, some of the reports are unverified. And so really, these are adverse health events that are supposed to be reported after following a vaccine, but some of them are unverified. And anyone can report an adverse event without providing a name or contact information. The advantage of having such a large database like this is that it allows researchers and scientists and the CDC specifically, to detect rare and serious adverse events quickly. So one of these examples are those rare blood clots that were appearing amongst those who are receiving the Johnson & Johnson COVID-19 vaccine. And of course, there were clinical trials that were very large, and they showed effectiveness and safety. But say, for example, the Pfizer trial had 40,000 people. So you may not be able to detect those very rare adverse events, like ones that are like 1 in a million. So VAERS can be a great way to identify some of those, and then go back and look at them carefully to really see if those are caused by the vaccine and if we should be worried. So VAERS is a great way to generate hypotheses for us to then go back and verify. The problem is when a database like VAERS is used to make causal claims. So Tucker Carlson and others have reported that, and this was a few months ago, he reported that there were over 3,000 deaths that were caused by the vaccine. The question really is, how do you make this claim from this kind of database? So let's say that all of the reporting is accurate and no one is making up the deaths that are being reported on VAERS. Still, the problem here is that, what are we comparing to? Are these deaths actually attributable to the vaccine, or would they have occurred anyway? So what would have happened if there was no vaccine in these same people? And that's how we actually demonstrate causality. So the first question to ask is, like, what are you comparing to? You can't just report the raw data that there were 3,362 deaths caused by the vaccine, because that's what's appearing in the database. Where's the comparison? So when we say that the vaccine is associated with, say, death or an adverse event, what we're really saying is that we're comparing a group of people who received the vaccine to a group of people who did not receive the vaccine. And then we compare their rates of these adverse events. And ideally, of course, you want the people who didn't receive the vaccine to be very similar to those who did receive the vaccine. So if those who received the vaccine in your study are much older than those who didn't, then naturally you might see more deaths amongst that population. But this is how you show association, by really comparing those, say, in VAERS versus those who didn't receive the vaccine. Causality is much more difficult to demonstrate because we're really thinking about a hypothetical scenario. Ideally, to prove causality, you want to show that everybody who received the vaccine, all of those in this darker shade, and you follow them over time and see what adverse events they had. And then if you could take this same group of people during the same time, not give them the vaccine, follow them over the same amount of time and see whether they had an adverse event. And if the rate is larger in those that are in the vaccinated group, then we can say that the vaccine causes a particular adverse event. How can we do this in reality? Because of course we can't have this hypothetical scenario of where you have the same people who receive the vaccine and the same people who don't. One way is to do a very carefully matched study where those who receive the vaccine are matched on all of the important variables or patient factors that potentially can affect their outcome and whether or not they get vaccinated. So you can design very well thought out studies that could potentially get at causality, making a lot of important assumptions. But this is how you show causality, not by reporting raw data from VAERS. So the next point I wanted to make is on the veracity and the reliability of real world data. So many of you may have seen these retractions that happened in two of the most prominent and most well-regarded medical journals, the Lancet and the New England Journal of Medicine. The one of them that was published in June 2020, actually the retraction happened in June 2020, was on hydroxychloroquine and COVID-19, and the other was on cardiovascular disease, drugs and mortality in COVID-19 patients. Both of these were retracted, which was a major deal because as soon as they were published, a lot of reports were shared on social media and in the media, regular media, talking about these findings. Some clinical trials were even planning to stop investigating hydroxychloroquine because of these studies, but then they were retracted. So just specifically this hydroxychloroquine study, which was published in May 2020, had looked at electronic health records of 96,000 patients in over 670 hospitals across 6 continents. So imagine the massive amount of data that they claim to have pulled together across all of these different countries. So as soon as these publications came out, researchers started questioning them. How did they get their hands on so much data so quickly? And so they quickly wrote a letter and sent it to the Lancet. Here, both of these papers, which were authored by several researchers at Harvard, relied on proprietary data that were analyzed from electronic health records that were provided to them by this company called Surgisphere, which is an American analytics healthcare company. The red flag was that this firm refused to make its raw data available to third-party auditors so that they could validate the results. This was a big red flag. But because of really the rush to publish these COVID papers, there wasn't as much thought in this peer review process that should have stopped these papers before they even went to press. So the lead author from the Brigham and Women's Hospital in Boston is quoted to have said, since we do not have the ability to verify the primary data or primary data source, I no longer have confidence in the origination and veracity of the data, nor the findings they have led to. So before they published these studies, the researchers and journalists should have asked more questions about how so comprehensive of a data set had been gathered from hospitals across the world in the middle of a pandemic. I've worked on studies where all we're trying to do is combine electronic health records across 2 different health systems within Pennsylvania. And it's a huge amount of work with lots of issues. We have to work carefully with computer scientists, informaticians, et cetera. So a bioethicist from the University of Sydney was quoted to say, this whole event is catastrophic. It's problematic for the journals involved. It's problematic for the integrity of science. It's problematic for medicine, and it's problematic for the notion of clinical trials and evidence generation. My final point, the fourth point, it has to do with reporting bias or sampling bias. And I really like this cartoon on the right, where they say that they received 500 responses and found that people love responding to surveys. Well, of course, the people responding are the ones that love it, and everyone else is tossing them in the bin. So similarly, some media reports, including Fox, have reported that only 1% of adverse events are reported to VAERS. So they're saying, look, these 3,000 plus deaths are only 1% of those that are being reported. So they have extrapolated to say that there are actually thousands of deaths due to the vaccine. But it's really more about what is being reported. When the 1% is stated, that means that it's 1% of major adverse events, but not minor adverse events. So for example, you could have a sore arm after a vaccine, and that, of course, is most likely not going to be reported. So when we say that 1%, really the reality is the 1% of the serious adverse events are being reported. And finally, cherry picking is an issue. So you may have seen this cartoon before on the right, where if you spin a wheel and really test a lot of hypotheses, you're going to find associations. So here, if you spin the wheel and you land on coffee, coffee can cause glaucoma or depression in twins. So this is just showing how if you do a lot of testing and if you do some cherry picking, you can really show a statistical association. So the example I have here is from the an Oxford study from the Evolutionary Ecology and Infectious Disease Group. And they published in a medRxiv in 2020, suggesting that half of the UK population had already been infected by March 19th, 2020. It was later shown by statisticians that their data source and their statistical methods to come up with that 50% number was incorrect. However, this came out and it was cherry picked by media outlets and created unnecessary confusion at a critical time because supporters of the passive herd immunity theory use this to say, look, we've almost reached herd immunity. So why do we need government public health restrictions? Why do we need a mask mandate? Why do we need social distancing? So it can be very dangerous to cherry pick results. Oftentimes this does happen to bolster political agendas. So I'm just going to wrap up here so we can get to questions. So what are the main things we should really be addressing before we believe real world data? We have to make sure that the data are true and they're a valid data source. The data integrity is really important. Can we generalize the results to the broader population, or are these specific to particular age groups? For example, can we reproduce the results? If your results are based on a small study, are you going to find the same thing in another study looking at a different population? There's also the problem of using raw data to make causal claims or even claims of association. So using VAERS, those are raw data. Instead, we should be relying on carefully designed and analyzed studies that deal with a lot of these statistical concepts like confounding and missing data patterns. And then also back to my point about the probabilities, we also want to be very careful about how we interpret results. Interpreting results correctly is key. Thank you very much. I like this, also like this cartoon on the right where it seems dramatic until you think about it for a second. So I'm happy to take questions now.
Okay, so thank you very much. Yeah, if you could cut your slideshow. We have one question, and if others have questions, submit them now or raise your hand. And I have a couple of questions for you also. You talked a lot about the VAERS system, the Vaccine Adverse Event Reporting System. FDA also has the Drug Adverse Event Reporting System, and it has, I believe it's called MAUDE, which is the one for medical devices. Mm-hmm. And these are all self-reported, and I think journalists are all often confounded by them because it's the source of information, but it has so many, caution flags with it. I mean, so two questions. Are these adverse event reporting systems ultimately helpful to the FDA regulators and scientists? And is there a responsible way for journalists to use the information?
Mm-hmm. I really feel that they are very important, but really most important just for regulators and CDC researchers and other scientists where when you start seeing that there are adverse events, it allows them to then realize that they should investigate this further. So it's as I mentioned, a great like hypothesis generator, because most clinical trials that are testing these vaccines or these medical devices aren't large enough to capture those really rare adverse events. So it's a very important resource for scientists. I would not really report any of these as having any type of association with the device or the vaccine from the media standpoint, because these all need to be adjudicated very carefully by scientists. So I feel pretty strongly that these data should be used only by the CDC or the FDA or other researchers who then can go and investigate further.
Okay, and I should also note the FDA's Vet Med division also has the same type of adverse event reporting system for animal drugs, which
right.
long time ago when I covered the agency, actually one of the more interesting stories I ever did was about a dog arthritis drug that was swamping the adverse event reporting system with lots of dog deaths.
Interesting.
So they can be used at times, but with huge caveats.
Yeah.
So I have a question here from Sandra Peters, who's a freelance journalist. How are health systems defined geographically? I'm wondering if randomized controlled trials take into consideration environmental confounds and control for variables like participants living in polluted locations.
Oh, that's such a great question. So health systems, generally, it depends on the data source that you're using. So for electronic health records, if they're using the same system, then they can potentially be linked pretty easily. So for example, in the University of Pennsylvania health system, there are some community hospitals that are part of the health system, and they're using the same REDCap database. And so those data can be easily linked, and we could look at them pretty carefully. But if different hospitals are using different databases, then they can be quite difficult to link. And sometimes there are also HIPAA concerns, like, is it okay for us to link these data knowing that we have patient identifiers? So there are a lot of statistical there's a lot of statistical research actually going into how to combine these data without having to look at the individual level patient data. In terms of looking at kind of environmental exposures, it's a really great question, because certainly we have seen differences in health outcomes in people who live in large urban areas that have access to world-class hospitals versus people living in rural areas where they may not have the access, but they're also potentially being exposed to other types of pollutants or other environmental exposures. So these are all things that potentially are confounders, variables that we oftentimes have to think about carefully when we're analyzing large observational data, but also we have to think about when we're designing a clinical trial too.
So for the journalist, a question here about how journalists should be interpreting studies that are based on randomized controlled trials or studies based on real-world evidence. Mm-hmm. What are the questions if a journalist is either looking at the latest FDA approval or looking at the latest article in JAMA, might be reporting out a RCT, it might be reporting out real-world evidence. What are the questions that the journalist should be asking of the scientist, of the people, of the study designers who put it together, so they can properly understand how the study was conducted?
So specifically for randomized trials, which are, have like very strict protocols and are usually very well designed, especially if they're appearing in journals like JAMA. One thing to just note, I think the biggest thing about randomized trials is, are those results generalizable to the broader population? So did the randomized trial restrict patients in any way? In the past, there were some randomized trials that were conducted, say, for example, to assess whether prostatectomy was effective in treating prostate cancer amongst men. But the population that was restricted in the clinical trials was only men below the age of 65. You might want to kind of just see whether there were any restrictions, some exclusion or inclusion criteria of the randomized trial that potentially may hinder generalizing to a different population, an older population, for example.
Now, you talked about the retractions. One was in the Lancet, and I think the other was in New England Journal. Mm-hmm. In the era of big data that we're in right now, is the temptation to kind of mine all these data that are out there going to lead to the, tempt researchers to go too far with the associations that they're seeing or they think they're seeing? Is because we have so much data now, is the temptation to cherry pick growing, or is it more than it used to be?
Yeah, I think, I don't know if the temptation to cherry pick has grown, but potentially, but I think there's a temptation to mine the vast amounts of data, as you mentioned, and to really, without thinking a priori, what am I testing? So really the principled way to do a study is to think ahead of time, what do I want to ask? What are the questions I want answered using these data, rather than just running, looking for any type of association that's coming out of the data and then reporting on it. So you're correct in the fact that like cherry picking results from large, like massive databases can become a much bigger problem when you have lots and lots of data without thinking very carefully at the beginning about what am I asking, how am I designing the study, who am I comparing to? Okay.
And so in the kind of the core question we're kind of raising today is, should real world evidence studies be used for drug and device approvals, either initial new drug applications or additional indications on drugs that are already approved? Mm-hmm. I mean, do you have a view about whether real world evidence studies can be designed in a way that makes them legitimate and beneficial in the drug or device approval process?
Yes, absolutely. I think it's an incredible resource, and it really the real world evidence. It's in the name. It's real world. It's showing exactly what's happening in practice to a very broad population of patients. And so it's really like a great resource, and it's critical for us to use real world data, but it has to be in a very principled way. You have to think very carefully about how you would analyze these data, how you would originally set up the study, and potentially look at the data so that you're emulating a randomized trial. Like if I were able to run a trial, how would I have done it with the data that I have? But it's extremely important to be looking at real world evidence.
Okay, and of the potential sources of real world evidence out there you talked about the electronic health record and gave the pros and cons of those. Are there, between the broadly defined electronic health record or insurance claims or, administrative claims like that are, gathered by public health departments, are there kinds of real world data that are better than others?
A lot of my work has been in cancer outcomes, and there are a couple of national databases that are very well put together. There's this SEER database that is looking at cancer outcomes, and there is also SEER Medicare linked database, and there's also the NCDB, the National Cancer Database. And so these are real world databases because they're really collecting data on treatment decisions, what treatments patients are getting for cancer, and what they are dying of, or are they having recurrences, all of that. Sometimes you can even link to the physician's data, like how much training did the physician have, et cetera. So these are really great sources of real world health data, cancer registries, and as you mentioned, also insurance claims databases. Okay. And with that, it's 12:45 now, so we need to draw this session to a close. Dr. Mitra, I want to thank you very much for your time and expertise on this.